Cool. Well, welcome everyone. So as I mentioned today, we're going to be doing our virtual owl pellet dissection. Some of you may have your own kits. If not, you're still welcome to follow along. I will be sharing. In fact, I'm going to stop my screen share. I'm going to, on this computer, if you want to pin it, there is a second Daniel Gillies video in the list, and my hand's on it right now, but it has the kit contents, which I can go through really quickly. So feel free to pin that if you like. Like I said, you don't have to leave your own personal video on if you don't want to. I'll be kind of working between two screens here. But the path for today is we've got the kit instructions, owl pellet guide and worksheet. So I'll walk through this together, and I'll try to pace it. So if anyone wants to stop or ask any questions along the way, they're welcome to. For a lot of us, this is the first time we've done this as well, which is kind of the fun part of the activity. In terms of what other things you will need today, of course, your owl pellet, shown on the dissection cam, wrapped up quite nicely. There's some nice tweezers that came with the kit, a little wooden probe. If you don't have these, just something really to take the owl pellet apart. A work surface. If you have the kits that we built, they come with paper plates. If you don't have paper plates, I just have some paper on my desk. It's really just for easy cleanup. They recommend, don't do the owl pellet dissection somewhere that you might be eating next. They are sanitized by going in basically a big oven. So they are sanitized, but it's just, taking that extra step of precaution. And also gloves. So I'm going to go ahead and put my gloves on so that I am ready to go. The kits come in two sizes. Some owl pellets are jumbo, some will be large. There may be a little bit of difference on what is inside the pellet. So actually, I'll be excited to see the differences in some of the kits that are out there in the wild. Okay. So starting with the instructions here, a little bit of background, and sorry if it's a little bit redundant for people. I'm just going to read right from the sheet. Learn about owls. Owls are classified as raptors or birds of prey because they actively hunt and eat other animals and small birds, the prey. Owls serve an important role in keeping ecosystems in balance by keeping the rodent population in check. It has been estimated that a family of owls, 2 parents and 4 young, can consume somewhere between 1,000 and 3,000 mice during the 75-day nesting period of the young owls. Owls often swallow prey whole, and the soft parts of the meal, such as muscle, are dissolved in the stomach before moving on to the intestine, where they are absorbed. The indigestible parts of prey, so that's the bones, teeth, fur, and feathers, are compressed in the gizzard to form an oval-shaped mass known as an owl pellet. All right, so really quick, if anyone doesn't know what a gizzard is, birds and a few other animals are specialized in that they have 2 ways of digestion. There's, like we have in our stomach, chemical digestion, and then in the case of owls and other birds, they actually have a mechanical form of digestion that basically breaks things up, and that's what's happening here. So continuing on, the pellet is regurgitated. So it is not poo. It is more of like a hairball. It is regurgitated back through the mouth about 12 hours after the prey was first consumed. So that's what we have here. Dissecting an owl pellet is a good way to learn about the eating habits and environments of birds of prey. Owls are opportunistic hunters and eat the prey that is easiest to capture. The larger the population of a particular animal, the easier it is for an owl to make a meal of that animal. Mice, voles, and shrews are generally found in large supply, and their remains are often found in owl pellets. But when their populations get low, owls will eat just about any other animal they can find, including rats, insects, rabbits, birds, and bats. And actually, in the case of a little lab group that I'm a part of, one of the other students had found the owls were actually eating salamanders, and not just that, but toxic salamanders, which was a bit of a mystery. And she just recently published a biological note on it. So there are still some pretty interesting things you can do in terms of science in studying owl pellets. A little bit of background, as I already mentioned. Safety note, this owl pellet has been sterilized to kill bacteria. However, we recommend that you still follow these safety guidelines. So for everyone playing along at home, never put owl pellets or parts of the owl pellet in your mouth. So no eating owl pellets. Work in a place separate from eating and food preparation areas. Use your disposable gloves like so. It's okay if they're too big. Just make sure that your hands are covered during the dissection and cleanup. Use a disposable plate to contain the owl pellet during dissection. So like I said, I just have a piece of paper to keep my work area clean, but if you have a plate, that is preferred. And thoroughly wash your hands, as everyone should be, especially nowadays. Work areas and any dissection tools when finished using a sanitizer. You can use household bleach in a 110 solution as well. So any questions before we get started? Good, because I might not know all the answers, but that's why we're going to do this together. So if you do have questions, don't worry. Feel free to ask them, or you can send them to us via the email invite, and we can answer them after, including from any students who might be doing this at home at a later date. So first, measure the length and width of the pellet using the ruler provided. So I'm going to do it here, and it's on my sheet. Kind of curious who's got the smallest and largest pellets out there. So mine, maybe a little bit hard to see on the screen, about 6 centimeters long. I'd say just about 4 centimeters wide. So I'm gonna write that down. Anyone else have any owl pellet data to share?
Yeah, mine was 5.8 centimeters long and 3 centimeters wide.
Anyone else? Measurements still in progress.
So mine was 4.5 centimeters long and 3 centimeters wide.
Do you have the large or the jumbo?
I have the jumbo.
Oh, interesting.
But I have two actually. I also have a 6.5 centimeter long and 2.5 centimeter wide one. They're both jumbo. So one's very long and the other one's just very wide.
Very nice. The next step, and I suspect most of the people don't have a scale, so feel free to copy off of mine, will be to weigh the owl pellets. So I'm going to start my scale up here. And then for everyone else, if you want to get started, you can start unwrapping your owl pellets on your plate, and we can head to step 3. I will take a quick measurement. So I have a jumbo pellet, and it's backwards on the screen, but it is at 9.5 grams, 9.5 grams. I'm going to write that down and record it. And then we can begin, if you haven't already, to start to peel away the foil. So Marie, your time has finally come. I know you've been waiting for this moment. There's my owl pellet. Any initial observations? Color. I'll share with mine first, and there are definitely some interesting things going on here. I'm going to try to bring it up to the camera. Mine's kind of a dark gray, and those look like, those could be teeth. They kind of look like teeth from a rodent. Then rotating around a little bit furry in terms of texture, kind of like matted fur, and falling apart in two spots. Doesn't really smell like much of anything to me. How about someone else?
In mine, I see quite a bit of fur, like clumped together, and it looks like the front bits of teeth too. Pretty dark brown and hard and doesn't smell.
Other observations?
I can go ahead and go. I have really dark brown. I can see kind of furry bits kind of all over. I have this light colored spot in here. And then almost looks right here, there's a a spot that almost, it's not teeth, it might be an eye socket kind of protruding.
Interesting.
Yeah.
Marie.
And so
I can share too. Mine has, I don't know if you can kind of see it there, but there's hay in it. I don't see any bones right off the bat, but there's definitely, like it was definitely in a barn. There's other like animal fur in here too. That's not, it's very wiry and straight, kind of more like a cat or a goat. Yeah, very interesting.
Interesting. Great observations. Anyone else who wants to share initial observations who has a kit with them right now? Oh, hey, Alex. All right. Well, I'm going to record mine if no one else has any other observations to share. Dark gray, matted fur, and no smell. Okay. Next steps, and I kind of, we've already done this a little bit. So look carefully at the outside of the pellet. Do you see any debris or particles on the pellet? They're not fur. I think Marie had a really interesting one, the hay and other type of barn materials there. What is on the outside of the pellet can give you valuable clues as to where the owl who regurgitated your pellet might take its home. And they give a few examples. So hay or straw, barn, exactly as Marie thought. Dirt or grass in or near trees and cut banks, pine needles in or near evergreen trees, grain, a grain elevator, and then seeds, a silo. Does anyone see anything different before we kind of go on to the next step? For mine, I'm making out just a lot of matted fur and what look like little bones and teeth. Don't see any hay, maybe some like little bits of stick that shows up. Get it to focus. There. Anyone else find anything different?
I think my light colored little group might be a little chunk of mud or dirt.
Ooh.
And then looking over, I do think I have a teeny bit of little grass kind of mixed throughout as well. So I would assume mine might be in or near trees and cut banks.
Cool. Anyone else? All right. Then the next part is an open question, and especially for any students that might do the lab at home. What do you think is inside the pellet? Describe your hypothesis in as much detail as you can. What kind, how many, how big, and why? And I think we've heard a few of those before. So on mine, I think I hypothesize that I'm seeing bits of a mouse or maybe a shrew, especially given the interesting teeth there and the amount of fur. How many? One, maybe, could be more than one elements of here, especially because there's kind of like two masses. And how big? I am actually not sure how big a shrew is, but in terms of a mouse, I'm thinking field mouse size, roughly. And why? Because it was dinner, and then these are the undigestible parts. I guess anyone else have any other interesting hypotheses or observations they want to share before we start to do the dissection itself? You guys looking intensely.
Hey, Daniel, just so you know, I don't have my kit. I only had one for me, so I'm saving it for class. So I'm listening and taking notes.
No problem. All right. All right. Well, I think we heard about some of the other potential hypotheses, like the barn environment from Marie earlier. So the fun part of this now is the dissection. So gather 3 paper plates or pieces of paper in my case, probe, here, tweezers here, a bone sorting chart, which they have on the worksheet. It'll actually help you in sorting the bones you find. And then the dichotomous key, which basically is a series of yes or no questions that you'll answer in order to identify what you found, or to help identify what you found. Got all that, check. Place your pellet on one of the paper plates. Place your other work areas nearby. These are going to be used to put any fur or bones on. You can also place the bones and fur into separate piles using the same plate. Using your fingers, tweezers, or probe, break off a piece of the pellet. Well, I'm going to break off a rather large piece. Starting with this small piece, well, we'll take a small piece of my large piece right there. I'm going to separate this out. Already beginning to get more fur details and what look like some smaller bones. There we go. Starting with this small piece, look for bones and other hard pieces that are stuck in the fur. If the pellet is really hard, you may soak the pellet in water to soften it. Rub fur between your thumb and index finger to feel for small bones and teeth still stuck in the fur. Place the bones on one plate and the fur on the other plate. So this process is basically going to continue. So we're going to use our tweezers and our probe and our fingers and water if we need it to begin to basically separate the bone out from the fur. And I'm going to read a little bit ahead while people kind of continue along, and then I'm going to join in. Then continue breaking pieces off the pellet. Be careful when removing fur from bones, especially skulls, as they may break easily, as they're fragile. After removing all the fur from the bones, sort the bones into piles, skulls, vertebrae, ribs, etc. Use the bone sorting chart to help you identify the different bones. The easiest way to identify the animals in your pellet is to use the skull dichotomous key. That's what we were looking at a second ago. After identifying the skull as a rodent, shrew, mole, or bird, it will be easier to identify the rest of the bones since they have to belong to the same animal. If you have two or more skulls that are not of the same species, just look at the bones in those categories to help you identify each bone. You'll most likely find the remains of rodents, especially mice and small birds, in your pellet. However, owls are not picky eaters, so you might find the remains of insects, bats, rabbits, frogs, or lizards. Discard the excess fur. So I'm going to get started here. Let me make sure I've pinned my video. There we go, spotlight for everyone. Okay, so I'm gonna start probing away. If anyone has any observations as they probe through, I'd be very interested in kind of hearing them out loud. Particularly Misty or Marie, what you've already been finding as you go.
So I actually found, and I guess like my observation is because I work with a lot of fish dissections, so finding something with teeth and a jaw intact is really cool. But I found like half of a jaw, and it's a rodent of some kind. It's got really nice carnivorous tooth here at the end that's a little bit overgrown. And like just like the teeth are all intact, which is just so cool. Like I didn't expect that, I guess.
Cool.
Yeah.
I think I just found something very similar. If I'll put it in my palm here too. So this is also a small jawbone. It looks like some sort of rodent. There are teeth. They're a little maybe hard to see in the video, but they're right there. And then there's a very large, a large tooth at the end, the type of teeth that you see kind of in the front. Those would be the incisors. Marie, I know you're more fish, but maybe you know your bones better than I do.
Nope. I think you're right. Incisor sounds right.
Sounds right. How about you, Misty? What have you found?
So far, I think it's like a little vertebrae. So it's so small, I'm not even gonna be able to show you, but it's like a little perfect circle, and I got 3 little notches kind of coming off of it. So like a little bit of a backbone.
So cool.
Very, very small and fine. And Oop, I'm not even sure. But maybe a little bit of a hip bone. Oh,
Oh, cool.
Mm.
Are you guys finding that the bones are really kind of tightly packed and kind of crunched together? Some of mine have been separated, but then I end up with a ball of small bones, presumably when it got crunched up through the gizzard.
yes, and they are very fragile.
Yeah.
Indeed.
So you have to go very slowly.
Mine I actually feel like are more spaced out, but I found like the ribs first, so maybe it's just that it was positioned a certain way.
Oh.
I don't know anything about bird ecology, but
I just found I have 2 different skulls in mine, which is cool. And there was one little packet. I thought it was all fur, and it's just the tiniest skull. I can show you. I think they're both rodent skulls. Let's see.
I'm gonna
So here's
bring you up to the spot.
the 2.
oh wow.
Yeah, they're tiny.
Wow.
But one of them's a
They're
lot
full.
smaller than the other. So
Wow. Do you think they're birds or rodents or?
I think they're rodents because rodents have 2 front teeth, like everybody knows the beaver, right? And then they have a big gap, and then they have their back teeth. So this guy has 2 front teeth and then some back like molars.
Cool.
And then this other one is tiny, so it must be like a baby shrew or something.
Wow.
Maybe a field mouse?
Yeah, could be.
Ooh.
Is anybody else finding just rocks? Like they're not bones, but rocks?
Really?
Yeah.
Little maybe kind of like tiny bits of them, perhaps.
Yeah.
So I know a lot of birds will actually digest small rocks and pebbles and sand in
Oh.
order to grind up the food in their gizzard because they don't have teeth like us. So they don't chew their food. They swallow it whole. And in that gizzard is where it will kind of, the muscles will kind of move it around and grind it up so that it can dissolve down better. So it could be a possibility that owls do the same thing.
Okay, yeah, I mean, I bet that's right then. Like bird behavior.
Any other interesting observations out there? I'm not sure all else who has kits, so I'm currently working through some large, might be part of a skull. Oh yeah, definitely. I can see the little teeth. A very fur-covered skull. So slow progress at getting it out of the fur mess.
Yeah.
Oop, there we go. Yeah, it's definitely a delicate process. So for anyone who's doing this for the first time at home today or in the future, take your time. Use the little wood probe very carefully because you don't want to break apart the bones that you're finding, or when you're working to put these back together, potentially to identify the full organism, if you can. It will be helpful to kind of keep them grouped together too. So not just purely all bones in a random pile. It may help to kind of keep them grouped the way you found them as well.
So Daniel, I think I have a leg bone. So just to talk about how fragile, if I can get closer, if y'all can see that.
Yeah.
I have, it looks like a little tibia.
Yeah.
Very, very itty bitty.
That's so
And
neat.
they're connected still. So that's fun.
Very cool. Yeah, as anyone calls out anything, I can change the spotlight to you so we make sure it gets recorded in the video as well.
Ah. So I'm not doing like a very good job of preserving it because it's so tiny, but I did find like an entire rib cage. But it's like falling apart, unfortunately.
Maybe the type of thing, if not that we have to do it here, but if you had a little a jar of water or like a bowl of water, maybe you could kind of dip it in and let it dissolve around it and see if the you
I
don't have
actually am gonna do that because I really want to see it put together. So I will be right back.
no problem. Please spotlight. So here's my skull finding. Let me, oh, wrong spotlight. We don't need me here. Ta-da. Far enough away that it shows up. So you can see the skull. The areas where the eyes would be. And then the upper part of the jaw, I'll get the tweezer. So there would be the front teeth, and then the upper part of the jaw on the top is actually right there below some fur. I'm just being careful so I don't knock the teeth off. That's kind of cool. All right. It is actually a very slow process to go through. More than I thought it would be. Although I'm also finding just large, uninterrupted chunks of fur. So I guess if you're like me and you have lots of chunks of fur, don't feel discouraged. Curious to see what Marie's water dissolving, how that works out.
One kind of immediate observation on that is that it's very buoyant, which I guess makes sense because it's probably like hollow bones, but just trying to get it wet, it's a little bit resistant.
Are you able to show it on the video?
Oof. God, I'm sorry. I'm still working on getting the fur off.
Oh.
I should have grabbed my dissection tools before I left my job. Yeah, but it's all wet now. It's kind of gross, but that's dissection for you.
Gross, but not dangerous to anyone. So
Exactly.
People, especially students who might be trying to follow along later, do not be shy or afraid. Can't hurt you. It's really not dirty. It's been processed and sterilized, so don't feel too grossed out. But
you know, and if you're a student that doesn't feel grossed out at all, that's not bad either. Like that's a good thing too.
Absolutely.
Yeah,
In
I was
fact,
that kind of kid.
And where has it led you?
I'm in Fish and Wildlife now.
Very cool. Actually, I guess while we're going through this, might be a good opportunity since there will be students who will be following along later. What is, for those in fisheries and wildlife, what's everyone's focus? What's their focus they're studying? And I'm going to pick on you first, Marie, because I still have you on the spotlight video.
So I'm really interested in limnology and specifically, I guess, like fish health. So fish are just really amazing creatures. By all accounts, salmon should not have lived as long as they have, in my opinion. But they're just really these incredible animals and they face so many obstacles. And one obstacle in particular, I guess that's happening a lot right now is just like a disease that's coming up from the south and it's making its way into Oregon's rivers and it's called whirling disease and it's lethal to the fish. So for me, what's really important is finding a way to work on, I guess, like policy and with other people to figure out how we can help stop this bacteria that's spreading. Anyway, that was kind of long-winded, but I like touching fish.
And actually, I'll ask you a follow-up because you mentioned doing fish dissection. So to draw a parallel here, what sort of fish dissections are you doing, and how does that help with the science?
Yeah, so I did fish dissections for a lab for a while, and it's very much like you can tell a lot about how healthy a fish is by how they look inside. And generally, if you take a sample of fish, it will tell you something about the population. So fish are kind of unique in that, like, they don't, I've never seen a fish kidney. I might be wrong here, but you look more at their spleen than their kidney for health. So it's just really interesting. And they also have, and we have it too, it's called an otolith, and it's inside your ear, and it's a little stone. And when you look at that stone, it can tell you how old a fish is and how much stress it's been under based on like the rings in it. So it's kind of like a tree. And that just kind of helps scientists understand what kind of stress populations are going under, and where the fish has been, because some water is saltier than other water. And it's just, it tells you a lot about the population, I guess. So it's really interesting.
That is an awesome description.
Thank you. Daniel, what about you? You're more wildlife. What are you working on?
I am an explorer in this field.
Don't forget to pin yourself.
Oh yeah, I will. I'm gonna pin my workspace. Let me do that. I'm starting to get some new discoveries here, and I will let you free. All right. So for me, my background's in engineering. So this is my second degree. So my first degree was in aerospace engineering, and now I am kind of exploring wildlife, but also have an interest kind of broadly in fish as well. So I've been trying to get exposure through the program at OSU to a variety of different subject areas, which, as I see more and more variety, it's showing me topics in ecology are actually pretty interesting, seeing how all of these systems interact together with animals and their environment, animals with their animals, animals and people, also known as anthropogenic sources of disturbance or change in the environment. So not doing any work with owls at the moment, although I just started volunteering at the National Aviary, which is a zoo here in Pittsburgh where I live. That is, ooh, I got a nice little skull too, starting to come together. Yeah, where they do have a few different species of owls there, and I actually did ask them about their owl pellets, which they do not save or donate. It just takes too much work to get them sterilized in mass quantities. But they're a really cool institution, and get kind of more direct exposure to some of the birds. I've been spending time at the zoo as well, working in the aquarium here locally, which has given me more exposure to fish, the systems that it takes to keep them alive at the zoo, but then also kind of understanding their feeding habits, how they interact with their keepers, which is really fascinating. You really, I think, don't appreciate exactly how much of a personality some of the fish can have.
Absolutely.
Yeah, I think there's so many questions and areas within wildlife that you probably, as a casual observer, just maybe don't even ever think about or consider. And then as you begin to explore it, it's amazing what's really going on in the natural world around you. So I don't know, that's a kind of a summary. How about I pass it to Misty, who is deep in one of her pellets. I'm going to put you on the spotlight as well.
All right, there we go. Sorry. Yes, mine pellets. I had a very crushed skull, so I'm like really doing some tedious work right now. My emphasis is really on conservation biology. So just to kind of go along a lot of what Daniel was saying, that behavioral ecology is what I really love, especially in our avian species. So I'm more towards the bird side of things. especially they are indicator species. So by studying the movements and behaviors of birds, you can kind of see the health of the environment around them, because if there's birds there, you usually have a healthy environment. They're one of the first things that you notice leave when you have an unhealthy environment. And so that can help to move a lot of conservation and know when we have problems in an area and what we need to start looking at. However, it's not just birds. They're kind of what I've been obsessed with lately, but I kind of like all of it, especially when it comes towards with conservation. It's really the education and the stewardship that goes along with it that I've been mainly focusing on. I do work at an aquarium. Our aquarium has mammals, fish, an aviary, so we have birds, and I get to talk about it and teach and how we can, what we can do when we go home to help kind of foster that sense of conservation in our everyday lives. So that's sort of where my focus is really. Conservation biology can take all different kinds of paths. I've also studied breeding birds and helped with banding. I've never worked with owls, so this is very exciting. But I have studied with nests and eggs out on the beaches locally where I live in Mississippi. So.
Awesome. Since you mentioned banding and we are doing a lab on birds, maybe for those who don't know a lot about it, just as another way of monitoring and the involvement of using banding in the research of birds, maybe could you explain what banding is and the importance, just like the importance of this today?
Yeah, so banding is basically when we have little bracelets on the legs of a bird, or maybe a tag on the wing or neck of larger birds. And so that way we can identify an individual out of a species, because all the birds look exactly the same. And we might have birds all over, but we want to study this one population in this one area. So we will put a band or a unique identifier for that bird so that we can study where that one bird moves around to, what it's eating, where it's nesting. Because once it flies away with all the other birds, we can't really tell them apart. So this is where scientists and biologists will use them to study and research where they can best apply conservation methods. Really, we don't know how we can help until we understand more about them. And so that's where banding comes into play. If you've ever been to a zoo or aquarium and they have birds there, you might notice they have little bracelets on their legs. They do that as well so that you can ID them, just like having a name tag on as a worker. We also do this out in the wild with populations of birds so that we can better understand them as well.
That's really neat. Shaylin, what do you, because you just got done with school, right? Almost. It's been a few years. But
Yeah, so I now work for the Fish and Wildlife Department, but before that, I did my undergraduate in zoology, and always wanted to work with wildlife and endangered species and do conservation work. And then I got my master's in conservation biology, and then started the last 5 years. I've been leading wildlife conservation study abroad programs mostly in Patagonia and in other parts of Latin America. And before COVID hit, I actually got to go work in Southern Africa too, which was awesome.
wow.
So now I'm back and focusing more on the mentorship and teaching side and trying to pass on some of the career tips I've learned over the years. But yeah, I've always had an interest in everything. I love birds and herps and frogs and snakes, and learning more about fish now too. So it's kind of the thing of, I think this field is you start out with one interest and by the end you love everything. I'm really into geology now too, so it just keeps building.
Awesome.
That's awesome, for sure. Like one thing I've gotten super into, because I started coming in with the idea of doing fish because I worked at a fish hatchery and I just love fish, but I've gotten super into bats. Like they're so cool as animals and just like in general because they're so like beneficial to everybody, but I think they get like a really bad rap. And so it's been fun just kind of like learning more about different kinds of bats and stuff too.
Checking in on findings. Any well, actually, I'm going to share. I'm going to pin this here. Hold on, guys. Oh, don't want to remove you from the spotlight. All right, so if I turn off my light, this might be a little bit easier to see. Not really. So I've got a whole collection here, and this is only from the mess of pellet. One half is still completely undissected. The rest is kind of scattered about, and I've been going and picking through my scatterings, trying to separate out the fur. And then this is my my bone collection. So I've got the large skull that I was picking at earlier, a smaller one that looks very similar, so probably also a rodent. Ones that simply haven't survived completely. They were crushed. So these are all like half of the upper jaw and skull and bits of the top of that one. I believe, and I'd be curious if people are finding the same thing. And this is not yet going to the dichotomous key or the sorting guide. But these smaller pieces, they look like ribs to me. They're very, very thin and curved, kind of a similar shape. In fact, looking at the sorting key, going to look like H here, which is marked as ribs. There's N, the sacrum, which I believe I have one right here. You kind of make out the shape with the gap in the center, and then just imagine a wishbone, basically, that type of shape. Then some of the leg bones and other elements of the arm just scattered. I haven't been able to sort them yet. So I don't know if anyone else wants to share their current findings. We could probably go through this for quite a while. So if there's any highlights, I will put you on the big screen. Otherwise,
so one of our pellets, we have actually uncovered 4 different, well, I don't know if they're different yet. I haven't analyzed it, but 4 skulls. So let me see if I can do this without spilling them.
Ooh.
There is 4 different ones here. And we were thinking we'd have 1. They are noticeably different sizes. So this is the largest one here. Gosh, it's hard to do this. And then noticeably smaller ones like this one. So it'll be interesting once we actually sort to see if they're all the same type of animal. But there's so many little ones, a lot of small ribs. There's more than I had hypothesized would be in here. So that's surprising to me.
Likewise. I am blown away every time I open a new ball of fur, and then it seems like there are a dozen ribs just all stacked together, and maybe they were together originally, presumably, although it's hard to tell sometimes.
Mm-hmm.
Any other items folks would like to share?
I have found 4 different scapulas These are kind of one of my favorite bones. I know that sounds weird, but they are really thin on top and I can't get like a good light on it. But yeah, they're really like kind of papery thin, like a bug. It's nice. They're cool looking.
This could be a very therapeutic activity in a way.
Right?
Through. Any questions from anyone out there? Or questions that students might have you think that we can attempt to answer today? And if not, we will get back to you.
Could be interesting. Maybe I can look it up and share a screen or something, but to look up what frog skulls look like or some of these alternatives to rodents. Include that in this video.
Any experts on the call who might know? I definitely do not.
I don't know what they look like. I do know that frogs are mostly made of cartilage because they have to function. And also like the bones would weigh them down if they're water frogs. But other than that, I've never seen a frog skull.
And they don't have teeth either, so that could be a big indicator.
Yeah.
I see. Well, perhaps you do a little research for us on that topic. I see, Alex, you've got a pretty good work area there. So I'm going to put you on spotlight if you don't mind. What have you found?
Sure. Can you hear me?
I can hear you.
All right. This is my first time using Zoom on a phone, so bear with me, everyone. Let me see. Okay. So I found some skulls here. What looks like a couple jaw bones, I think. This guy. A lot of jaw bones and ribs, and then don't know bones that well besides that, but I assume these are probably leg bones or something. I keep looking for an easy one, like a hip bone or something like that, but I just know head and maybe a limb, but a lot of that. So yeah, I think these are shrews or something, or I don't know, or maybe they're mice, but I guess I never thought of how big a mouse skull would be, but maybe too small. Or
we can actually, since you've got it up there on the screen, this is a good opportunity to use the dichotomous key for owl pellet skulls. So you keep it there. I'll read through the key. 1, does the skull have a beak and lack teeth? If yes, it's a bird. If no, go to step 2. So you're finding?
Yeah, I'm gonna go with not a bird.
Not a bird. So no beak, and it has teeth, correct?
Yes.
All right, so we're gonna go to step 2. So
Okay.
next, does the skull have a diastema? And that's a large gap between the front and back teeth. And that would be the gap between the back teeth, kind of on the upper part of the skull, and then those front incisors that we were looking at.
I'm gonna go with yes.
All right. Yes. Go to step 6. 6. Is there a small peg-like tooth directly behind the incisor? And yes, the incisors are the teeth located at the very front of the mouth. So small peg-like tooth.
I'm not seeing one, I don't think, because it's kind of just a space here,
Okay.
I think.
Looking at some of my examples here as a comparison, and I don't see any either. All right, so no small peg-like tooth directly behind the incisor, so go to step 7. 7, this one's a little bit invasive. Pull out a back molar. Does the tooth have a root? And then I'll keep reading it. If the tooth looks like a long tube, is the same color from top to bottom, and does not split into points at the bottom, then there is no root. So a root just like our teeth. It is not unusual for 5 or 6 teeth to come out attached to each other when pulling out a molar. It's probably really hard. They're very small.
Ooh.
Yeah.
Does it say why?
It does not say why.
That's interesting.
The world's smallest dentist to pull that one off.
If anyone else has one and wants to try it, I don't know if Alex, if you're able to get it out or not, maybe poke it with the end of the wooden stick.
Well, I do have this garage tool that's more dentist-like, so.
Sure. Improvisation is perfectly fine.
Yeah.
I know in mine, they're pretty loose on mine, so I'm able to kind of push at them. Doesn't take too much force. Oh yeah, that's a great view. Well, yours are really in there.
Oh, there we go.
All right. Is it a long tube in the same color from top to bottom?
I don't know how well you can see it, but I don't think so.
Okay, so not a long tube. So if no, go to step 9. Do the back teeth form triangles?
I actually have to go to the dentist in like 3 weeks, so this is making me a little nervous. but I'll do it for science. No, I'm gonna go with no.
Okay. Are there 4 to 5 separate back teeth? We're now at step 10. We've made it all the way to the bottom of the key.
Oh, geez.
Four to 5 separate back teeth.
I think that looks like 4 to me, kind of 1, 2, 3. I can confidently say 4.
All right. Then the key says it is a pocket gopher. Class, it is a mammal in the order Rodentia, family Geomidae.
How about that? Oh, so maybe it's a pocket gopher family sharing the same pellet in the end. Because that kind of, oh no, this looks very different actually.
Yeah.
This other one.
So I think that's a good question for students that are doing this to consider too, is who gets made lunch? Is it like what part of the population gets to be lunch for the owl? Because in general, it's probably going to be the slower ones, because those are the ones that the owls can get to.
And the little guys, the young are definitely going to be easy targets.
Yeah.
Yeah, with kids, you could talk about too how owls hunt. They have really incredible ability to muffle their own sound, and also an incredible hearing ability. So like we call them facial discs on owls, how like the barn owl have a heart-shaped face. That allows them to funnel sound best to their ears. And so they can hear things that are even under the ground or under the snow, and they'll just pinpoint it exactly and dive down, and their wings are so quiet that the mouse, underground doesn't even hear the owl before they're captured. So that might be pretty cool to students.
Yeah. So I actually have a question for teachers here. Have your students ever dissected anything before? Have you guys done any other kinds of dissections? No? Okay, cool.
This really is a good starting dissection, that's for sure. Not quite the same as experience as, say, a frog or something like that, but lots of discovery, and you get a variety of different potential species in one dissection. I got another little skull.
For sure. My first dissection was a goat, so this is like a lot more fun than that.
Wow, that is intense.
Yeah. I guess like perks of growing up in like a farming area. Yeah, there are goats everywhere.
All right, I've got a little skull, so I'm gonna try out the dichotomous key on it. I'm gonna add myself to the spotlight here. Everyone together. Hard to see, so you'll have to take my word for it. So one, does the skull have a beak and lack teeth? It definitely has teeth, so no. Step 2, does the skull have a distema? Large gap between the front and back teeth. Well, it has incisors and big back teeth, so yes, it does have a diastema. So go to step 6. Is there a small peg-like tooth directly behind the incisor? So I'm looking at mine very closely. In fact, I'll just do it here. I do not see any small peg-like tooth. So no. Step 7, pull out a back molar, the dental procedure again. And does the tooth have a root? Let's see. These are very, interesting. On mine, try to share it as well because it came out as a section of like 3 triangular teeth. It's gonna be really hard to show. Well, I'll describe it. It's tube-like, that's for sure. They're kind of like a zigzag in pattern. Obviously very small, generally white, but they're not one color. They get very dark at the bottom. It does look like a long tube, but not one long tube. Is it the same color? No. Does it split into points at the bottom? Possibly, hard to say. So on mine, it definitely is not the same color from top to bottom, that's for sure. It says it's not unusual 5 to 6 teeth to come out attached to each other when pulling out a molar. These look like one continuous tooth, actually, although it may just be the points at the bottom. So I'm going to say, does the tooth have a root? Yes, on mine. Is the skull longer than 25 millimeters? Step 8. So there's a little measurement, and I have to find the skull on my work area here. And it is definitely not longer than 25 millimeters. My skull here is, let's see, 13 centimeters. So, sorry, 1.3 centimeters, 13 millimeters. So no, then it is a mouse. Genus, moose. Moose. It's fun. So my small mouse skull.
Very cute.
Anyone else have anything interesting that they want to go through in the key?
Yes,
or,
I do.
Oh, awesome.
Not for the key, but this was really kind of jarring is that this one, I gotta try and get a better light on it. This mouse was not digested. There's his eye and
Whoa.
there is fur. I was pulling off like chunks of fur and I was like, oh, that's kind of weird. But the other side you can kind of see is a skull. And
Wow.
then we've got his little eyeball. Poor little thing.
That is wild.
I kind of feel bad for the owl too. He didn't even get like the really fatty part of the animal.
Is it definitely a mouse?
It is not. I need to like uncover the skull, but it's very, very fragile. So I'm trying to be very careful.
I don't know if this is the same. So like at the end of my pellet, it's almost like the outside is coated. Is that what?
Kind of like really thick and like hard to break.
Uh-huh.
Yeah, mine was that way too.
And and that's maybe this is a more of a mouse than I looked like at first glance. based on how the fur is coming up, it's hard to tell where it's attached and where it's not, but I think I might have a partial version of what you've got.
Ooh. So one really cool thing that you can kind of see on this mouse too, and for students, maybe if you have a pet rodent, like I had hamsters when I was in 3rd grade, is that this mouse was not chewing on anything to down its teeth. So they got really long and it's really hard to see, but yeah, they just keep growing if they don't chew anything. So this mouse, its teeth were just wild.
Whoa. I had a skull, but it collapsed.
Oh no.
So Marie, when you say not chewing on anything, do you mean it didn't eat or it was just really soft stuff?
I think probably like softer things. Like again, this is based on my experience of having hamsters, is that like you have to give them like wood and like salt to chew on to break their teeth down. Otherwise they like their teeth can grow like up into their head. So I don't know really how that works for like field mice or other mice, but I think it's the same system because beavers have that too, I believe.
Yeah, I definitely have something similar to what you had. It was the one, I'm gonna put it on, I'll spotlight it.
Oh yeah.
So similar to yours, there's the teeth. And then I was like, oh, it just seems like really matted fur. But now I'm realizing like, oh no. If you look at that all together, you can kind of see that it wasn't just matted fur. It was definitely fur matted to the skull.
Yeah.
It all kind of came out together. And I'm not going to take this one apart for the same reason. It's kind of interesting like that.
Right.
Cool. So a variety of things that you will find in different states of digestion. I guess if depending when it made the pellet, maybe some are further along than others. So maybe you have to make a balance between what the owl's able to completely use. But I'm not sure.
One thing I think would be really cool, maybe is less plausible for students, but to kind of like cast these in resin. You can get resin at your local Joann's and you can just put your little skulls in it. Keep them forever.
Make little dioramas and
yeah, or like make a little necklace or some earrings.
I always kind of wondered when I saw there's people who have arts and crafts that have little bits of these bits. Maybe this is where they get them from.
Probably. I know, I was thrilled the first time I did this when I was a kid. They like didn't let us keep the bones. And I'm like, now I get to keep them. I'm so excited.
We've got a few notes on chat people have had to run so far. So just for those who did have to go, thank you, because you'll see this on the recording at least. So thank you for attending, and hopefully the steps that we walk through are helpful for everyone. And it looks like we still have Kim. Don't feel obliged to hang on the whole time, as you will have the recording since you're not going through one of the kits. But we'll just kind of continue here together and probably work through trying to identify some of the other bones, and then we'll call it a night. So thank you for joining us.
Thank you.
All right, so the 4 of us, I'm gonna pin us all to spotlight.
Have we looked at a hip bone?
No. Here,
L.
you pull up the hip
Number
bone.
L is pelvic bone is the official term.
So I'll show
I
you.
just uncovered one, super small. You can see there is that rounder part.
Ooh.
It looks just like the picture.
Yeah.
Awesome.
Very cool.
Can y'all, is that focusable? I think so. Yep. I have a lot of jawbones, but my owl waited much longer than your owls to expel his pellet. They are very condensed, crunched, and fully digested.
So it makes me wonder if like digestion functions change as the owl maybe gets older, or maybe depending on what like sex they are, if they're male or female.
It could also be that mine, when did we say how often did they, every 12 hours about? So what if the last half of the 12 hours mine didn't find any food, so yours was more well fed?
Right.
True.
Yes.
Yeah, especially since mine had hay in it. I'm sure that owl was eating good.
So mine could have came from an area with less prey
sustenance.
populations.
Wow. I feel like every time I just move something, a bone appears. So now it's more of just sorting through the remnants for bones. Has anyone tried overlaying the bones on the diagram on the sorting chart? It might be kind of cool. See how far you can get. And then you can kind of be a bit of a paleontologist as well.
Oh, this is really cool. I'm probably gonna break it getting the fur off, but I have like a leg bone that's connected still.
Wow.
It's got the little joint attachment.
That is cool. I want to hold on before you. I'm going to put you pinning in the spotlight. Let's take a second.
Okay, no worries.
Love to share that. All right, go for it.
All right, this little guy's got his joint still attached. So that's a little tendon. And it looks like this is the tibula. So the arm bone, or no, this is the ulna and the radius. Yeah, so it is the arm, but it's the ulna and the radius, and then the no, not the femur. Is it the humerus? Yeah, so I think this one's the humerus and this one's the ulna and the femur.
Oh, cool. But no scapula. It doesn't go up that far.
No, my scapula was not attached.
See, has anyone found any scapulas in their bones?
Yeah. Here's one.
Nope. I'm gonna pin you up.
Yeah, use your tweezers and hold it up.
Oh, let me get my tweezers. There we go. That's so much easier. Use the tools you have.
Very clear. Nice.
Wow. Oh.
We also have, where is that backbone, little vertebrae? All right, so one thing we've learned is better sorting from the beginning.
Is important.
We thought one paper plate would be okay and we could do our 3 piles instead of 3 paper plates because we wanted to conserve, and not always the best idea. So this little guy is N and either J or O. So N is your sacrum, so your tailbone area. And
there we go.
Oh, wow.
And then that's either going to be part of the vertebrae of his backbone or the tail. So
That is so cool.
So Jeremiah thinks it's tail and I think it's backbone. So that's a nice hypothesis we're going to figure out later when we line up our bones on the diagram.
Yeah, I think things to emphasize for anyone watching this who haven't started it yet. Have plates. One, have lots of plates. It is a messier activity than it might look, and it's dry messiness, so things just kind of scatter. And early
I would-
organization is key.
Yep. I would say even just layer the table with maybe newspaper.
Yeah. I think another big thing is if you're like a heavy breather, you might want to wear your mask for this.
Indeed.
That way it doesn't
Yes.
get up your nose or like all over your table.
Suppose if you have an allergy to fur.
Yeah, my nose itches so bad right now.
Oh no.
Oh, what is this? I think this is a pelvic bone. Has a kind of a hole. It's not continuous. I think maybe part of it just maybe snapped, but I'm pretty sure that's a pelvic bone. Looks like a pelvic bone.
Oh, that looks really big from here. I see it big, but
If I get myself out of scale, it looks. I got the super jumbo.
actually, scale, that's a really good point. I'm gonna be right back. I have a sheep skull here and I'm gonna hold it up right next to the mouse skull, just kind of give like a little extra layer of kind of like size reference. So this is the skull of a sheep. You can see, like, it's got these really nice crunching. It's got crunching bones, or it's got teeth for vegetation, so they're not sharp and pointy like other teeth are. And you see how big it is. And then this is a field mouse.
Amazing.
Very tiny, and also carnivorous because it's got nice little pointy teeth. All right.
Is this the one we think is the red?
All right. How far along are folks?
I don't know.
Oh, I'm probably in like the last third of mine.
Yeah.
I'm down to very small bits.
Me too.
Just lots of itty bitty ribs, and mine was pretty thoroughly digested and small bits. I have most of the lower jaws of mine are all in two pieces.
Mm-hmm.
And I have like 1, 2, 3, 4, 5, 6, 7, 8. I have at least 8 lower jaws here. So 4 fragmented skulls.
Yeah, I'm gonna do a count here too. Let me see. I've got from the. I guess. Oh, does anyone have a bottom half of a skull? I feel like everything I have is a top, and I don't want. I have one that might have it here, but I don't want to.
This here's the jaws. I went with that.
I do have like a bottom jaw, but I
Those
don't
are the
have
ones
the
that work to
bottom
make
of the skull.
I think it was just dirt holding them together. Or,
Yeah, so each of my, the bottom jaws are sort of like this. So each side, because most all skulls are not connected all the way across in a lot of animals. So they're easy to come apart. But you can see the teeth, you can see the bone that comes up and attaches up here by the, I guess, the ears of the rodents. That's my largest one. I think that one went to my pocket gopher. Pretty sure that's what this guy is.
Oh, whoa. So
Hey, Daniel, thank you for letting me hang out with you guys. I will leave you a note. We're doing our kits on Friday, so I'll leave you a note on it.
Awesome. And if you get a screen cap or anything from the session you do, or if you have, I don't know if there's any kids in person, send it along. That'd be awesome.
You got it.
Thank you for coming, Tim.
Hey, thank you so much. You guys are great. Take care. Bye-bye.
Bye. All right. So following on what you were just describing, Misty, it made me pull away part of my partial skull. And now it's very, very visible. What you were describing in one piece, though. So here, there was the fur covered side that almost looks like a mummified head. And then on the back side, you can see the upper part of the skull is here. There's that lower jaw, and it's still all being held together by just the fur that it's kind of clinging it together. So I'm not going to pull it, but one of the teeth, and I'm kind of poking it right here from the bottom, and it comes up and kind of has a bit of an underbite, or it just got crushed into that. It's really cool.
One thing
Where's
I've
the
definitely
diagram?
learned doing this is that I'm really glad I don't have a gizzard. This just looks terrible. Like all the little bones, because mine was a barn owl, so I do think it was mostly just eating like little mice. So like there's just like hundreds of ribs in here. Yeah.
I would say I wonder how it feels to them, but I guess it's just life. It's a normal process. So it probably
Right?
feels like, what would you mean we don't have to make a pellet? That's such a crazy
They're
concept.
probably thinking like, gosh, I'm so glad I don't have like a digestive tract and a rectum. Like,
Those chemical digesters are so strange.
right.
I mean, they do. It's just downstream. They've got two-stage digestion.
Oh.
I mean, they still have to chemically digest. It's just so all the other bits happen downstream of the gizzard.
Okay. So that might be a good thing to like make a note of too, because that's a question. Again, I'm not a bird person, but so they do, you said it's not poo, but they do still poo, right?
Oh yeah, absolutely.
Okay.
All birds still, well, what's the book? Everybody poops.
Everybody poops. Yeah.
Everybody poops, even owls poop, but this is not owl poop.
Okay.
Say
It's actually feces and urine mixed together.
Oh, so
all
kind of like
in
hummingbirds.
one.
Yeah.
Mhm.
Okay.
So what they do digest, those small muscles and tissues that are digested, still go out the same track as anyone else. But the bones and everything that we would usually eat around that they swallow whole, they kind of pick through it. And fun fact, some fish-eating birds actually swallow feathers to make like a mesh to keep the bones from going through long enough for them to completely digest.
Huh.
And so it kind of slows it down. And so it's like a feather like plug in their gizzard for the bigger stuff to have more time before it goes through, because the fish eating ones, like a horned grebe, don't have pellets.
Oh, that's neat.
So,
That's really cool.
Here's I'm gonna go add spotlight. So there is a digestive tract of, in this case, it is just a reference digestive tract of a bird. So from the top, the esophagus and crop. So our food makes its way in, then down the proventriculus. And I can give a little bit of definition. Proventriculus. They don't define it here. That's not fair. But it contracts and helps the food move its way down. And if I remember correctly, proventriculus starts chemical digestion slightly. Let me take a look. I've got my textbook here. I am 90% sure, but I wish I had the full definition here in the book. Yes, the proventriculus is the glandular area of the stomach that produces the mucus, pepsin, and hydrochloric acid necessary for digestion. Note the ridges inside. Then the gizzard, which is where the pellet is being made, is just below it. So where my finger is pointing to.
Okay.
It has a heart-like shape. I will give an exact definition from the book: the tough muscular section of the stomach that acts as the chewing teeth for toothless birds, grinding food material into a soft pulp and acting as a safety valve to block the passage of bones and other objects that might injure the soft intestinal tissues.
Huh.
So this
Dang,
is,
I kind of do wish I had a gizzard then. That would make things a lot easier.
the Manual of Ornithology, obviously backwards in the video, but this is one of our textbooks. This was I think Systematics uses the Manual of Ornithology, and Ornithology uses the other book that we have.
The other ornithology book.
Probably called Systematics. Good book though, actually, a really great reference if you want to learn more about kind of college level of avian structure and function.
Yeah, that's good to know.
So Alex, I know you came because of your sister who is in another lab with me.
That's right.
So what brought her to volunteer you for our lab?
You know, I don't know. She just said, you're going to get a thing in the mail and do this. And I'm like, okay, Jane, that sounds good to me. That sounds like fun. So here I am.
Thanks for joining us.
Yeah.
I can't remember what she said you work on during the day when you're not dissecting owl pellets.
I actually work for a locomotive manufacturer. So very different from everyone else on here. But
That's
at
really
night,
cool.
at nights, I dissect owl pellets. By day, trains.
Awesome.
So, yeah.
That's really cool. So like, do you have like I guess like a favorite train to work with, or like, how did you get into that?
So I actually don't work on the shop floor. But I like to give tours in final assembly building because it's really easy to tell what part of the train it is. Like, yeah, that's the wheel. Can't you tell by the
Right,
part?
right.
And that kind of thing. No, so I'm more in the sourcing and logistics part, but kind of worked out over time. Summer jobs and then jobs after college sort of worked into it that way. So
Neat.
it's good though. Always need trains.
Right. Best way to transfer something.
Mm-hmm.
Very cool. You need to guilt her. Is she not fisheries and wildlife? Am I imagining it? I mean, we're not in a fisheries and wildlife lab separately. It's water chemistry, but
Yeah.
I swear.
She is fisheries. I don't know exactly what she's going for, but a lot of diving and marine biology she's been doing lately. So close.
Cool. Well,
That's
you should
cool.
still guilt her for not participating directly.
Yeah. I said, like, Jane, I doubt you had better plans tonight than this. So whatever excuse wasn't as good. No, I have been trying to convince my wife. So I had a bird feeder right outside my window growing up. So that's kind of what got me started. And then now I'm at 1, 2, 3. I think we have like 5 or 6 bird feeders on our property now. So she's trying to stop me. So that's why I'm like, Jane, all right, get me this for my birthday next year. Then it'll be too late. You know, once we have it, we'll just set it up and keep going. So.
I know the feeling. I just got my 3rd bird feeder today, and my partner was like, "You have to stop." He was like, "You bring another bird feeder home."
Yeah, you're supposed to say, "I'll never stop. You can't make me." Is what I
Right,
say.
exactly. So you can't tell me what to do. You're not my mom.
Yeah.
We were looking at, I said, no, don't get it. That's way too expensive. It keeps popping up on my Facebook. Maybe you guys have the same, except not you, Marie, because I know you don't use Facebook. A bird feeder with a webcam built in, and it's supposed to do automatic bird identification. It's a Kickstarter, and it keeps popping up and popping up and popping up, but it's $150. Like,
That sounds so cool, though.
It does sound cool, but it's like I could probably get a normal bird feeder and a webcam and put them together.
Yeah, I was thinking about getting just like a game trail cam or something, which would more or less do the same thing.
Yeah.
That would be cool.
I know we have one that just sends pictures of whatever it takes, like, to our cell phones. for game trail cams.
Oh, cool.
Yeah.
So I have a rat skull.
Ooh.
It's a small one, so maybe a juvenile, but this is the rat.
Oh, nice.
So I did follow the key. It
Mhm.
did have roots on its molars, and it was just over the size that it needed to be. So
That's awesome.
it's
Look
little-
out.
I think if I do this again, I'm gonna get a smaller pellet. Like I got the jumbo one because I was like, I want to get at least like a few bones, and now I'm like, okay.
So I saw the disclaimer that there might not be anything in the pellet.
Right?
So I got the jumbo as well. We got two jumbos. And Jeremiah already said, did they have smaller ones for you to choose? And I'm just like, that's such a silly question.
I have something I can't identify.
Ooh.
And it's going to be hard to show, but I'll try anyway. So that's the shape. I mean, it doesn't show up very clearly. Maybe there. I can kind of see it. I'll get myself out of the way. Oh, I'll do that. So if I were to describe it, it almost looks like looking at a crustacean shell, in that it's actually a bit rounded. Rounded, comes to a point at one end, which I could almost convince myself it's a lower part of a jaw, but it doesn't really have that form or shape unless it's something very, very young, which is possible. It kind of wraps around. Wish I could get a better picture of it.
Could it possibly be the back of a skull?
The back, I don't know.
Because the skull is in multiple pieces. If you look at the sorting chart, the top left, skull and jaw,
Mm-hmm.
it's kind of zoomed in and you can see how you have multiple, and I keep finding my crushed heads individual pieces. Like I even had a round, like the back area where your spine comes through the
Mhm.
skull. It is literally just a small, I don't know if that's even focusing.
Huh, yeah.
Yeah.
And it's just where the backbone has the hole to go for your spinal cord to go up into the brain stem area. So maybe you have a back part.
It might be. It could be a little bit crushed in on itself. 'Cause it's weird how it forms a point, but maybe that's like nesting into the skull in a particular area.
Is it maybe like N, the sacrum?
No, it's got. I have a sacrum here. There's no hole through it, but it is kind of hollow in a sense.
Okay. And is it thin like the scapula, or is it kind of like thicker material, I guess, like one of the bones?
I accidentally crushed it, so maybe I'll find out by looking
So
inside.
thin.
It was thin. Nothing else that would help me identify it other than it almost formed like oh no, like the body of an insect. Oh, maybe it's the body of an insect.
Oh,
That is possible.
it could be. I had a worm in mine, like the outer shell of like a maggot almost, or something similar.
If an ant's head was very large, it would have been like that. So it could have,
Like a cicada?
possibly some sort of maybe insect head. Probably my best guess. I'm going to take my. I think I'm hitting the end of my usable pile. And for anyone who's doing this on the video, I think you're going to have a lot to work with, probably more than you have space. So I think once you're able to identify kind of each one of the different types of bones on the bone sorting chart, and you've been able to work through the dichotomous key, then I would say you've successfully achieved the purpose of the lab. There are a few other optional activities, which I'll just identify here really quickly. After removing all the fur from the bones, soak the bones in diluted bleach. If you're a student, do this with an adult or have an adult do this for you. Soak the bones in diluted bleach for 3 minutes to whiten them. Use white glue to mount the bones you found on the bone chart or on index cards. If using index cards or cardstock, label each card with the type of bone mounted on it. It's a cool activity. Optionally, the bones can be placed and labeled in a display case for better viewing and presentation. They have a very specific one recommended. Try to reconstruct a whole skeleton from the bones found in your pellet. Lay the bones on a piece of paper and use the bone sorting chart to put the bones in the correct places. Or finally, make a food-based chain on the animals found in the pellet. That is a very good activity. It would take a little bit of work. I'm going to start to fold up mine here as it's beginning to become unmanageable.
I want to encourage students to, like, if you can get outdoors and take an adult with you and go exploring. Owls are really common, honestly, and especially in like the Albany, Scioto area. Like all you have to do is really listen for them, and maybe if you go out exploring safely, maybe you'll find your own pellet to make with an adult.
Just remember, if you do find an owl pellet to dissect out on your walks, you want to take extra precaution and maybe even look up how to disinfect it with the heat.
Yes.
Because it will be not as sanitized as these.
Agreed.
Yeah, and unfortunately animals have like a bunch of diseases that they can pass to us.
I'm going to be right back while I take this away.
Salmonella is a big one we want to be cautious with when it comes to birds.
Yeah.
Not just when we eat chicken, but also in their waste products. I'm not sure if that includes pellets, but if these were, my face itches now and my hands are dirty. If these were sanitized ahead of time, you know, there is always a risk for something we want to be careful about.
All right, I'm going to show off my bone assortment. I have not put them in the order on the chart, but kind of a wide variety by size here, from a variety of bones throughout the skeleton, the skulls, some that still have the fur attached to them, as we saw earlier. Really a very cool assortment. So I'm going to put this away in a bag for this one. I might actually try the index card mounting activity on another night, because this is kind of cool. Especially when it comes time to take systematics of mammals or anything like that.
Right.
I'm just putting my folded up paper with the bones on a plate. For anyone who also is contemplating it, definitely follow the instructions of using gloves. They were extremely helpful throughout this process. And make sure when you are done to disinfect your work surface. As I'm sure even if you use the paper plate, there is the potential for a bit of a mess. So
Yes. And when you wash your hands, make sure you're washing up to like your elbows, probably, because like for me, I have stuff all the way up to my elbow, so I'll need to wash really well. It gets a little messy.
here, I thank you guys for participating into this tonight and for all the great questions along the way. It has been recorded, so what I'm planning to do is I'll save the video and I'll probably do a little bit of editing so that there's some sped up version when we get to the kind of key parts and people are just listening to all of our chatter. But yeah, I think if you guys want to keep going on Zoom, I'm happy to leave this session up for you all. I actually have some exams tonight, ichthyology, and then plant genetics to finish up tonight. So
Good luck with the ichthyology exam.
thank you. I'm pretty sure if I make one of you the host while you continue to go through, it won't close the meeting.
Okay, I do think I'm gonna leave too. I need to go get something to eat. So
Yeah, if
with all you.
Probably gonna head to bed as well.
Yeah.
three of us are on the East Coast.
Yes.
All right. Well, thanks guys. I really appreciate you
Right.
joining.
Yeah, thanks. It was fun.
Yeah.
Thank you.
See you.
Bye.